Thank you. Our next presentation is S.Biomedics, and I'd like to welcome Sale Tony King, President and CEO, to the stage.
Hi, good afternoon. Thank you for your attention and coming to listen to about what we do. So we're very excited to be here to share our commitments on targeting Parkinson's disease through our programs. And please be advised that statements in this presentation are forward-looking and may vary. So today I'm going to give you a brief introduction of our company, followed by one of our leading programs, TED-A9, which is a cell medicine for Parkinson's disease. We are a cell medicine company based in Korea, and through 2 cell engineering platforms, we're currently developing 7 cell medicine programs. And out of the 7, 4 are currently under clinical trials. And the backbone of the company are these 2 cell engineering platforms. And one on the left, our first technology is called TED, which differentiates any sort of pluripotent stem cells into neural precursors at high purity, therefore giving us an access to target CNS diseases. And on the right, FACS is our second platform technology that enhances cells' function and forms these cells into spheroids consistently. And through the 2 technologies, we are currently developing 7 programs, and out of the 7, 4 in purple are under clinical stage, which includes Parkinson's disease, spinal cord injury, critical limb ischemia, and crow's feet. We recently finished our critical limb ischemia phase 1/2A study showing very promising results, and we're expecting to enter next phase by 2025. We also do have a very solid and concrete IP portfolio with 154 applied and 102 granted globally. So now is the intro of our TED-A9, which is our program for Parkinson's disease. And the slides that I'm going to cover are going to cover most of the data from our in vitro and in vivo studies, as well as initial data from our clinical studies as well. So I'm not going to delve into the specifics of what Parkinson's disease is, but I'll focus more on our efforts and current work. First, here's the development scheme of our TED-A9, which was published in Cell Stem Cell this year. We believe that embryonic stem cell-derived ventral midbrain dopaminergic precursor transplantation is a promising therapeutic strategy for Parkinson's disease. So to support this, we developed a robust clinical-grade differentiation protocol to secure ventral midbrain dopaminergic precursors. So today I'll be talking a lot about the ventral midbrain dopaminergic precursors. And for us, using only 4 small molecules, we can differentiate embryonic stem cells into ventral midbrain dopaminergic precursors in 25 days, while maintaining master and working cell banks throughout the production process. As I said, the final product is the ventral midbrain dopaminergic precursors, which matures into dopaminergic neurons after direct administration into the brain. Our standout achievement is that we successfully activated the signaling pathways leading to ventral midbrain dopaminergic precursors by using only 4 small molecules. So unlike the previously reported methods that relied on recombinant proteins for differentiation, our approach exclusively utilizes 4 small molecules only. And additionally, at each phase of differentiation, we have observed high levels of expression of appropriate markers, further validating our methods. So going into the purity of our differentiated cells that are secured under our protocol express ventral midbrain dopaminergic precursor markers, which are INGRL1, FOXA2, LMX1AB at all above 99%. And I would really like to point out that this is the highest EO ever reported so far. And as a cell medicine currently under clinical trial, we have implemented a master and working cell bank in our production process. The transition from our working cell bank to the final product takes 6 days. And throughout this period, we observed that the expression of STM progenitor markers was barely detectable, and the levels of early-stage neural precursor markers were very low and gradually decreased over time. So these characterizations indicate that our final product is composed of pure ventral midbrain dopaminergic precursors. Our final product, the precursors, matures into ventral midbrain dopaminergic neurons. And on the left, our study demonstrates that these mature cells express NER1, PITX3, and TH, confirming that they are indeed ventral midbrain dopaminergic neurons. On the right, we show that these mature cells secrete dopamine, further validating their identity. So we can conclude that we have successfully secured ventral midbrain specific dopaminergic precursors that matures into neurons which produce the appropriate levels of dopamine to target Parkinson's disease. In our in vivo study with rats, we observed significant behavior recovery by week 16. As shown on the right, the models injected with ventral midbrain specific cells demonstrated greater recovery compared to those injected with non-specific dopamine cells. This underscores the critical role of ventral midbrain specific cells in addressing Parkinson's disease. Our histology analysis at week 16 shows that the transplanted cells expressed high levels of PITX3 and TH. This is a strong evidence that the transplanted precursors successfully engrafted and matured into ventral midbrain dopaminergic neurons. So I'm not going to get to details about into the details, but one particularly intriguing finding is the composition of our cell population. By analyzing TET-A9 cell population, we observed that the transplanted cells express higher levels of GERK2 compared to carbidins. So this suggests that our cell population contains a higher proportion of A9s than A8s or A10s, which could be a key differentiating factor for our treatment. Our STR and PET analysis provide strong evidence that the transplanted precursors successfully engrafted and matured into functional neurons. The PET data reveal increased dopamine synthesis activity, indicating that the transplanted cells are operating effectively. Additionally, HPLC analysis shows a significant increase in dopamine concentration in the striatum. So together, these findings support the underlying mechanism of our treatment. This slide highlights the relationship between the number of transplanted cells and recovery outcomes in our in vivo study, emphasizing a clear drug dose response. A key takeaway from this research is that it provided a foundation for determining dosage in our clinical study. So as illustrated in the graph, the minimum number of cells necessary for recovery was 10,000. So when we convert this figure to human equivalents, it translates to approximately 1.87 million cells per hemisphere, totaling around 3.7 million cells for the entire brain. Consequently, we established our clinical dosing parameters, setting the low dose at 3.15 million and the high dose at 6.3 million cells. Our second key piece of in vivo data demonstrates significant behavior recovery in our primate study. So we created a Parkinson's model by administering MPTP to 6 marmosets. The increase of the score represents the increase of the Parkinson's behavior. The blue line indicates the 3 non-treated marmoset score, which remain unchanged from baseline, suggesting no improvement in their behavior. In contrast, the 3 treated marmosets, represented by the pink line, exhibited a remarkable improvement starting at week 6. By week 28, they had returned to normal behavior, supporting the hypothesis that TET-A9 effectively addressed the symptoms of Parkinson's disease in these animals. The findings of our marmoset PET data align closely with those from our rat studies, indicating a successful translation from smaller to larger animal models. The marmoset data demonstrate that transplanted TET-A9 were effectively engrafted and released dopamine as confirmed by PET imaging. And also notably, we observed a progressive increase in dopamine detection over time as well. And these results provide a strong evidence for the mechanisms behind behavior recovery. So our marmoset study not only elucidates the mechanism of the treatment, but also highlights its promising potential in targeting Parkinson's disease. So here's a video illustrating the change of marmoset behavior over time at 2, 4, 10 weeks post-treatment. The top row features a non-treated marmoset with no changes in behavior, indicating a persistence of Parkinson's symptoms. In contrast, the treated marmoset in the bottom row demonstrates significant improvement starting from week 6, And as you can see, by week 10, the marmosets show a sign of recovery, approaching normal behavior levels. So this visual evidence reinforces the effectiveness of our treatment approach. All in vivo safety studies show that no tumor and toxic effect were observed, and there was no issue with cell distribution as well. So now going to our clinical study phase 1/2A. Our phase 1/2A study is underway in Korea with a total of 12 patients. The 12 patients are divided into 6 low-dose and 6 high-dose patients. The participants must be 5 years of post-diagnosis and have responded to dopamine medications but are experiencing motor complications. Our total evaluating period is 2 years post-TET-A9 transplant. Our key evaluating factors in efficacy are FPCIT PET, which not only measures dopamine release, but also assesses intake levels, which is an essential component to our mechanism, and MDS-UPDRS Part 3 off, and Hoehn-Yahr scale, which both evaluate movement behavior of patients. So although our evaluation period is 2 years, this is an open label study enabling us to release midterm data at 1 year post-TET-A9 transplantation. So as mentioned, phase 1/2A participants were divided into low and high dose groups and were further divided into 4 different groups depending on the transplant period. As shown, the first group of 3 participants received TET-A9 transplants in May 2023. The next group was treated in September 2023, with additional transplants occurring in January and February of this year. We have transplanted our TET-A9s to all 12 patients within 1 year, and until today, we have seen no safety issue from any participants. So in terms of efficacy, one of the key parameters we're using is the MDS-UPDRS Part 3 off, which a lot of our competitors are using as well, which involves physician-recorded assessments of motor functions. This scale allows us to analyze the behavior in patients when they are not on medication, providing valuable insights into treatment's impact on Parkinson's symptoms. A higher score indicates more severe Parkinson's behaviors. Our first group of low-dose 3 patients were transplanted in May 2023, allowing us to evaluate data after 1 year. So initially, the baseline average score of the patients were 61.7. We're looking at the top row of 3 patients, indicating a relatively critical stage of the disease. After 12 months, 12 months post-transplantation, the average score decreased to 49, representing a reduction of 12.7 points. So although this is our initial data package, we're very excited because the data is an evidence providing that our TET-A9 actually did reverse the ongoing pathway of Parkinson's disease. So we're very excited to evaluate and review our next participants group, the high-dose group, in early part of next month. And by April 25, we do expect post-1 year analysis of all 12 patients. Our clinical PET data study continues to support the translation from animal models to human applications and further confirms our underlying mechanisms. We're utilizing FPCIT PET, which not only detects dopamine release levels, but also identifies synapses formed by transplanted cells. As these cells establish a synapses and release dopamine, we expect the uptake to increase. The color scale ranging from blue to green to yellow to red indicates the higher uptake levels. So before the transplant area predominantly showed a blue and green colors, whereas 1 year post-transplant, the area has transitioned to green and yellow and even red, showing the uptake increase. This data demonstrates that TET-A9 cells were successfully engrafted and formed synapses, which provides strong evidence of our proposed mechanism of action. So what we are trying to say is from our rat to monk primates to human applications, we do have a very strong translation data. So coming back to our development scheme, we have secured highly purified ventral midbrain dopaminergic precursors by using only 4 small molecules, which was not done before, which are expressing key markers at all above 99%. And furthermore, the transplanted precursors matured and expressed NER1, PITX3, and TH, which are expression markers of ventral midbrain dopaminergic neurons, which eventually secrete the needed dopamines for Parkinson's disease. So to summarize, we believe that we have secured medicine to attack Parkinson's disease and applied the program in GMP settings for production and secured exceptional in vivo results, both in efficacy and safety, and transplanted to all patients in phase 1/2A of the study. So although we are at the initial stage of releasing some sort of promising level of clinical data, we are very excited from the early package outcomes at this moment, and hopefully be able to share advanced clinical data in the near future. Thank you.